Hello boys, welcome back to another session of video tutorials, and this session is meant for class 11th biology students, and this is for 3rd December 2020. As all of us know, we are into the revision mode. We are revising the entire syllabus, and as part of it we are revising chapter number 13, Photosynthesis in higher plants. And in our previous session we talked about the Kelvin cycle, how C3 gets fixed. So today in this session we are going to talk about C4 pathway, carbon 4 pathway. It's also called Hatch-Slack pathway. This pathway was worked out by Hatch and Slack 1965 and 1967. Mainly operational in plants growing in dry tropical region like maize, sugarcane and sorghum. In this pathway, first stable product is 4 carbon compound. That's why the name C4 pathway. There we called C3 because there it is 3 carbon compound, right? Here it is OAA, oxaloacetic acid, OAA, oxaloacetic acid, and so called as C4 pathway. C4 plants have a special anatomy, internal structures. So Cranch anatomy, it's called Cranch, named after the scientist who discovered it. Vascular bundles, what is Cranch anatomy? Vascular bundles are surrounded by bundle sheath cells arranged in a wrap-like manner. It's a covering. It's characterized by large number of chloroplasts. Chloroplast number will be more. Thick wall, very thick wall, impervious to gases, and absence of intercellular spaces. That means there is no flexibility when the intercellular spaces are absent. It is tight. The primary CO2 acceptor is 3 carbon molecule. What is that? PEP, phosphoenol pyruvate, PEP, present in Mesophil cells. Remember? Mesophil tissue. And enzyme involved is PEP carboxylase, PEP carboxylase. Oxaloacetic acid formed in Mesophil cell forms 4 carbon compound like malic acid or aspartic acid, which is transported to bundle sheath cells. In bundle sheath cells, it is broken into CO2 and 3 carbon molecule. Because it is 4 carbon compound, remember, it can be broken into 3 plus 1, 3 carbon molecule and CO2. The 3 carbon molecule is returned back to Mesophil cells to form PEP, phosphoenol pyruvate. The CO2 molecules released in bundle sheath cells enters to Kelvin cycle. This is important. Where enzyme rubisco is present, and because of that rubisco, sugars will be formed. So the C3 and C4 are thus interconnected, and C4 in a way is helping C3 cycle. So there are 2 types, and what is that we said? So you got a first Mesophil cell, this green one, Mesophil tissue, because chlorophyll will be there, and this is a bundle sheath cell. So there are 2 cells, and these are very tight. We said cell wall will be very thick, plasma membrane, no spaces will be there, and then between this plasmodesmata will be there, small what you call channels or canals will be there, so that materials can get exchanged. So basically it's atmospheric CO2. This atmospheric CO2 is absorbed by the Mesophil cell. So HCO3 and phosphoenol pyruvate, PEP. So this gets fixed. So C4 acid will be formed. Oxaloacetic acid OAA, that gets transported through plasmodesmata to bundle sheath. Again it is the C4 acid. So decarboxylation takes place. That C4 will be broken into C1 plus C3, and that C1 is nothing but CO2, which will be entering into the Kelvin cycle and gets fixed. And because of this CO2, with the help of Rubisco getting fixed through Kelvin cycle, forms sugars. And the other C3 acid gets transported from bundle sheath, enters back into the mesophil cell, and there it helps in regeneration of PEP, phosphoenol pyruvate. So this is what in general we can say, in nutshell we can say. And as all of us know, photorespiration, another concept. It is the light, that's why the name photo, light dependent process of oxygenation of RUBP and release of carbon dioxide by photosynthetic organs of plants. Normally what happens in photosynthesis, we have said CO2 plus H2O, both are absorbed, and we get starch, or rather plants get starch plus some amount of water gets released, and finally oxygen gets released, which we wanted. So here it is getting fixed, CO2. Now in photorespiration, what is happening? Release of carbon dioxide, because it is respiration by photosynthetic organic organs of plants. Photorespiration decreases the rate of photosynthesis when oxygen concentration is increased from 2 to 3% to 21%. See, when oxygen concentration increases, photorespiration decreases. Presence of light and higher concentration of oxygen results in the binding of Rubisco enzyme with O2. This is the main crux of this. Rubisco enzyme combines with oxygen. And what happens when this happens? Rubisco combines with oxygen, PGA is formed, and plus phosphoglycolate. This pathway involves chloroplast, peroxisome and mitochondria. 3 organelles are involved. So photorespiration do not occur in C4 plants. Photorespiration occurs only in C3. Now what is the difference between C3 and C4 plants? For a layman, of course, all the plants are plants only. But for us, being biology students, we know that there are 2 types of plants based on the carbon. One is a C3 plant, another is C4 plants, based on the whether it is a C3 compound or C4 compound which is formed. So C3 plants, they don't have any special anatomy. We said cranch anatomy. We just now saw the picture also. Whereas in C4, there is a cranch anatomy in the leaves. Photorespiration occurs here. PGA will be formed. Rubisco will be there. Here, it does not occur. Photorespiration does not occur. PGA is the first stable product, and here PEP is the first acceptor of a phosphoenol pyruvate, is the first acceptor of CO2. And plants are adapted to all climates, C3. Of course, plants are adapted to tropical climate. Of course, OAA, oxaloacetic, is the first stable product in C4, and C4 plants are adapted to tropical climate. In the beginning only we said. And mesophil cells perform complete photosynthesis. Here, mesophil cells perform only initial fixation, that's all. Afterwards, they require cranch anatomy. So these are the broadly 2 differences between the C3 plants and C4 plants. Now, what are the factors affecting photosynthesis? As we guess, expect, light is the first factor. As light intensity increases, the rate of photosynthesis also increases until light saturation point. Once the saturation point is, then decrease. That's why we said, remember, 680 to 700 nanometers, and beyond that or less than that, there will be a rate of photosynthesis decreases. So light is a must. You can graphically also represent this. Carbon dioxide concentration. With increase in concentration of carbon dioxide, rate of photosynthesis increase till compensation point. Again, you can graphically also represent this. Temperature. It does not influence the rate of photosynthesis directly, but at higher temperatures, all the enzymes will get denatured. So once the enzymes get denatured, enzymatic activity will be inhibited. And once the enzymatic activity gets inhibited, naturally rate of photosynthesis will be definitely affected. And as we said, is inhibited due to denaturation of enzymes which affect the dark reaction, that carbon dioxide fixation. Blackman reaction will be definitely affected. And then most important, water, we said photolysis. Due to increase in amount of water, rate of photosynthesis does not increase proportionally, as after saturation no more water is required during photosynthesis. Only the plants require limited amount of water. There is no need for excess water to be made available to the plants. And there is what is called Blackman's law of limiting factor states. If a chemical process is affected by more than one factor, like we said, four factors we listed which will affect photosynthesis: light, CO2 concentration, temperature, water. If a chemical process, photosynthesis is a chemical process, is affected by more than one factor, let's say light and carbon dioxide, then its rate will be determined by the factor which is nearest to its minimal value. That means out of the light and carbon dioxide, which is the nearest to minimal value, obviously it is the carbon dioxide concentration. So that will decide the limiting factors. It is the factor which directly affects the process if its quantity is changed. Suppose we change the carbon dioxide concentration. Will the entire chemical reaction changes? Yes, definitely it changes. So that is what Blackman's law of limiting factors. And I think with this we have come to a conclusion. We have completely revised this chapter. I want you to write a small notes on that. And if you got any doubts, please post it in our